Okay. So this is going to be the first of a series of talks centering on the principal subjects in cortex, which are listed here. We have stem cell biology and cortical development. We have large-scale network dynamics and perception, behavior, neuroeconomics, repair, remediation, and training. The teams involved are listed here. I'm not going to go plodding my way through here, but you can see at a glance it includes the 3 major neuroscience institutes in Lyon, plus a group working in genetics, physiology, and molecular cellular biology, which has got the team of Valérie Castellani, and the group over in the Koenig's group, which is called the EMC, Olivier Koenig, and last but very much not least, the neuroeconomics group. And this is an important aspect of the Cortex project because the University of Lyon is very keen to have an integration of the social sciences and the biological sciences. And the inclusion of Marie-Claire Vival's group in neuroeconomics, who works with the group of Jean-René Duhamel and Dorea, is an important aspect of the justification for our existence. So, so far, Cortex has actually been a huge amount of agony because we've had to decide what we're going to do with 11.5 million euros, which you can imagine scientists can spend hours and days and weeks pondering that delicate question. And that's what we've been doing for the last year, in fact. We've been doing that. Today, I hope, is going to be the beginning of something much more interesting, where we actually will meet together and show our determination to build bridges between the different communities in neuroscience in Lyon, and to go towards developing something which is more than the part, more than the different aspects of the part. And I think that to do this, it requires a real effort on our parts, it requires a real sacrifice, it requires an investment, and this is what these cortical lectures are going to be about. It's going to be in a first series, we hope to have more, getting the principal investigators of the 15 teams that make up Cortex to talk about their research, their aspects of research which they find principally interesting. So because we want this to be the fundamentals of a common knowledge, we're filming these talks, and we're going to put it either on the web or on the internet. That depends on the sensitivity of the principal investigator. But the aim of it is that for those of you who can't be at a particular conference, it will be possible for you to look at this and thereby to build up this common knowledge of our expertise in Lyon, in the domain of neuroscience, which is really quite tremendous. So this is about building a community. There are, of course, well-defined communities in existence in Lyon already, but what we want to do now is to bring those communities together on a very focused series of themes, which is going to be covered by the Cortex Consortium. So today, I'm going to talk and Mr. Dorea, I don't know where you are. Perhaps you could act as chairman. Make sure that I don't speak over my time. I always do. So you have to take this microphone from me. Tell me when I've done 35 minutes, and then tell me when I've done 40 minutes, and then 45 minutes you take the microphone away, and I stop. So I'm going to be speaking I should speak for 35 minutes, then maybe a few questions, then we have a break, and then Mr. Dreyer will be presenting his group. So I'm going to be talking about what my team does, which is cortical connectivity. This team is interested in understanding how information flows through the cortex, looking at the structural constraints which exist in the cortex. What we have here is a well-known view, I'm sure you've never seen it before, of the major cortical areas as were defined by Brodmann. And the common philosophy is that these areas have a particular cytoarchitecture, that have a distinctive physiology and a set of connections. And if we understand the way these areas are interconnected amongst themselves, we will come to an understanding of how information flows through the cortex. My talk is entitled The Strength of Weak Links in the High-Density Interareal Cortical Network. And this is from my team, which is headed by myself and Ken Knobloch. This is a photograph of Ken, for those of you who haven't had the delight of meeting him in person. He's not here tonight. But Ken plays a very central role, and I hope you'll see where that role comes, because we're looking at some rather sophisticated statistical analysis of the cortical network, and Ken is a major figure in the translation of our rather rudimentary understanding at the anatomical level into these more sophisticated. Now, the title actually comes from a paper in the social sciences, which was The Strength of Weak Ties. This paper has been cited 21,000 times. That's about 10 times more than David and Feldman Van Essen's hierarchy paper in 1991, which is a sort of landmark. And basically what this paper is saying is that when this paper came out in the 70s, there was a feeling that there was something missing in the social sciences in understanding how local groups integrate into the large picture. And I think this is exactly the situation that we're confronted in the understanding of the cortex, is to understand the large-scale model of the cortex and the local connectivity and seeing how these two actually interrelate. Well, I began to get convinced that this was going to be an issue of weak connections when in 1980 with Pascal Baron and others, we showed that there was a direct connection between the primary auditory cortex and the primary visual cortex, area 17. Now, one definition of a primary area is it doesn't receive and interconnect with other primary areas. Well, they do. This connection actually is highly selective because it goes to the peripheral representation of the primary visual area. But nevertheless, it is extremely interesting because the group at the time working with Helen Girard and others at and Jacques Pernier's lab were interested in visual auditory integration. And they were having evidence that this was happening at a very early stage. And this pathway was clearly possibly implemented in that. And so if you have meaningful weak connections, statistically significant connections between area V1 and area A1, you might expect to find them elsewhere, and indeed that happens to be the case. So that the talk I'm going to be giving is going to be very much talking in a first instance about the density of the cortical graph, and I'll show that raises some intriguing issues about how a dense graph is going to actually have any kind of selectivity to it. I'll be showing you that the cortical graph has a density of 70%, near 70%. That means that 70% of the connections that can exist do exist. So if somebody says to you, oh, your pet area, LIP, and some other place, area 46, is connected, well, if it's a 70% connectivity, that binary evidence of connections is perhaps not going to be as useful as you might have thought. So that raises some problems. It raises some problems for the notion of a small world that I'll talk about. And I'll show you that despite this high density, surprisingly, there are some definite binary features which are rather selective, actually. The second part of my talk is going to be actually the meat of it, which is that this rather bleak thing of 70% connected is in some way put into a perspective, because it turns out that there's a very, very high heterogeneity in strengths of connections between areas. And there lies the important information, because I believe that it's there that we're going to be finding a selectivity and an important aspect of the cortical architecture. And indeed, I'll be showing you that this principle is highly predictive. It enables us to predict many, many important features of the cortical graph, and that's an important part of my talk. I'm going to be covering too much, so this is why Mr. Dre is going to have to rip the microphone away from me, because actually the interesting theories about how the cortex function enormously turn around notions of hierarchy. So I really do want to say something about hierarchy, because I beg to differ on many of the ideas that have been put about cortical hierarchy. What I'll actually be trying to get to is it turns out that the connections, the broadband connections, the connections which can actually drive target areas in the cortex are mostly between areas on the same or adjacent level in the cortical hierarchy. The other connections are far too weak. They're long distance, they're very selective in a certain sense, and I'll be talking about that, but they're far too weak to be actually driving in the classical sense of the word. So those of you who think and worry about cortical hierarchy, of course, know the idea that the forward-directed projections are driving and the back projections are modulatory. I don't think that's true, and I'll be chipping away at that block a little bit. So a lot of the weight of this talk is coming from a database which we've finally been able to publish, and it's got 1.5 football teams involved in the authorship of this paper. Some of them I'll be directly naming, but they've all played an incredibly important part. It's taken some 12, 15 years to amass this body of data, and it's now published, and we'll be publishing, or in the process of publishing, the models and more theoretical concepts which come out of that. The key players here, the people that remain from one generation of students to the other, are the three engineers which are here. There's Pierre Misery on the far left, and Camille Lamy in the middle, and of course Pascal Joux. I don't know if she's here, but she actually runs the team, and she seems to run most of the institute most of the time. So she actually is a key figure in all of this. And Pierre and Camille have put an enormous amount of work into archiving the connectivity, and it really is it's great to be working with these people. So, of course, this is what you probably all imagine I'm doing, or if you don't, you probably think this is what I should be doing. Because this is modern, it requires an expensive piece of machinery, and it makes wonderful pictures. These pictures are absolutely stupendous. They're usually in rainbow colors, and they enable you at a glance to understand the full connectivity of the brain, and the human brain to boot. So we're all very homocentric, and understanding the whole of the human brain at a glance in rainbow colors is exactly what we want. The problem with the technique is, and it's worth remembering, is the technique doesn't go down to the cellular level. And these lines, these beautiful lines, are not axons. They're probabilities of diffusion of water. And those axons, or those probabilities, do not go into the cortical gray matter. Now that's important because it means that you don't know if A is projecting to B or if B is projecting to A. It doesn't give you any sense of directionality. Now our findings is that the connections are highly directed. They're highly directed and they're not symmetrical. So saying that every connection in the brain is bidirectional is not going to work. So there's two problems here. There's a series of problems here that means that this technique is not suitable for doing what I want to do. Because it doesn't go into the cortical gray matter, all our understanding of hierarchy is based on the location of cell bodies and their terminals, in particular layers. That's what we mean by hierarchy. That's what we mean by hierarchy since the word go. This goes back 40 years. And it's basically an understanding of the connectivity of the cortex at a large scale, yes, but at the cellular level. And you can't look at hierarchy with DTI at the moment. Maybe in some future generation of these machines you can, but at the moment that's certainly not the case. It doesn't give you a signal in the gray matter. And the signal itself gives you a resolution of something in the order of 1 millimeter. Now, how many axons do you get in a millimeter? I don't know, but what I'll be showing you is that the range of strengths go over 5 orders of magnitude. And a lot of these long-distance connections, which are undoubtedly playing an important role, will not be picked up in any meaningful fashion by this technology. So you have to do what we do, and what we do has been done for a number of years. By the way, a lot of this work has been done by Nicholas Markov. He could be giving this talk. He can't. He's in America doing his postdoc training, but he did a huge amount of the anatomy and a lot of the modeling. So we're looking at retrograde transport. You inject a tracer in a target area. the tracer is transported considerable distances into the source area where it labels the cells. And if you want to have an understanding of the number of cells, then you count up the number of cells that are retrogradely labeled, and this gives you a handle on the connectivity. Now, when you do this in area V1, you get, these are the number of areas that you get connected. That's actually 25. So that's a huge number compared to what people knew about. And in red, and in all the slides I'll be showing, red is a color code for connections which hadn't been previously reported. So the sand-colored connections are the known connections, and the red are connections which are new. So what I'm going to be telling you is that actually, in this paper that we published last month, 30% of the connections that we can detect in a graph of 29 areas have not been seen before. They haven't been recorded in any sense. They're unknown connections. And I think they have a very important role to play. So this was largely done by Ana Rita Gomez, who's here today. So it's looking at the connections at distances from where we've injected all 29 areas. I've injected 29 areas, or we've injected 29 areas. And sand-colored is the known connectivity, and the red is the color-coded for the newfound projections. And what it shows you is that these newfound projections are relatively rare at short distances and become predominantly more and more prevalent as the distance increases. People have been looking under the lamppost. They've been interested in the visual system. They stab and inject a visual area. They look at other visual areas, and lo and behold, they find connections, and those are the ones that they know about. If you're interested in the visual area, you might not look at the auditory area. You might miss the auditory projection onto area V1 after so many, maybe hundreds of investigations of the connectivity of V1, simply because it's not part of where you expect to find connections. But if you look farther afield, you do find them, and you find these are the connections we find. They also happen to be weaker, because we will be coming back to this. But as you move away from the injection site, and this is on the left-hand side, as you move away, the connections become weaker. There's a relationship between strength of connections and the distance of the connection, so that the newfound connections tend to be weaker, and they tend to be at greater distances. So if you take the 1,615 connections that our 29 areas have, and you look at the number of, you make a graph out of that, so you make a square matrix, and you look at what is the frequency of connectivity. This is what is defined as the density of the graph. This is what is plotted here, and this is our density, and it's 67%. When I was saying earlier on, you have 67%. That's 60% of the connections that can exist that do exist. That's what it comes down to. Now, if you look at the average shortest path, it gives you an average shortest path between any two areas of about 1.5 steps. That's very, very short in the fully connected graph that we're talking about. If you remove randomly connections, as you remove the connections, the density of the graph goes from 67% down to 1%, and you get an increase of the path length going from 1.5 up to 3. That's to say 3 hops. That's sort of interesting, because now we can look at the previous databases which have been used by people to make profound statements about the large-scale models of the cortex. Now, basically, the earlier study was done by the study we all know, I think you all know it, which has been used enormously, referred back to as the study of Feldman and Van Essen. They looked at a total of 32 areas, and they reported a density of 32%. Now, other people have come back to their database. This report here by Honey et al. In PNNS, and Young in 1993, and others going up to this year have come back to Feldman and Van Essen's database, and they do something very strange. The connections that hadn't been tested by David Van Essen and his student Feldman in 1991, they say don't exist. And by doing that, and by adding on areas in a sort of higgledy-piggledy fashion, they end up with densities as low as 25% and 15%. At those densities, the statistics of the graph is completely different. So these are studies which want to understand the fundamental properties of the cortical network. They're taking a database which exists, and they're helping themselves to what happens to be there. And they're saying, if it hasn't been tested, the connection's not there. That's a pretty extraordinary way of doing things, because actually when David Van Essen published this paper with a density, an observed density of 32%, his prediction was that the density would be actually 45. He makes a prediction in this paper. It's one of these, you know, it's 40 pages or something in cerebral cortex, and of course nobody reads that much. It's, you know, reading has gone out of fashion altogether. But if you read it, it's clear. He's saying, I think this is a 45% density. And by the way, David Van Essen is one of our collaborators, so this is not a ding-dong thing. I don't have a disagreement about that. At that time, Jouve, shortly after, in around about 1998, updated the known connectivity and came to a point of something like this, about 48%. And he used a graph theoretical technique to make a prediction, and he predicted that the complete density would be about 58%. So we're not very far from what we're observing here at 67%. Now, these papers here, and more papers which have been published this year, I could have them here, but they're not, including Nature Neuroscience, have come to the conclusion that the cortex has a very definite principle in its organization of the network, and that is that it conforms to a small world. The small world network is a network which has, which is very well understood, very familiar. I don't know if it's very well understood, but it's very familiar. It's the idea that your 6 handshakes from bin Laden or the Queen of England or whoever it is, and that this is possible because of a mathematical property of that social network, and that is that the average shortest path length is actually surprisingly short, and the clustering, that is to say, the connectivity amongst the adjacent nodes is very short. So your friends all know each other, and you know 1 or 2 people in Africa, and they have a tight community, and it's this combination of clustering plus very rare connections going across the cortical graph, which leads to this notion of a small world. And of course, it's very attractive to neurobiologists understanding the cortex, because a small world gives you the possibility of segregation and integration. It seems to address this issue that I referred to at the beginning of my talk about how do you incorporate the local into the global. Now, if you look at if you increase the density, so this actually, this green line here is at the density where the path length is 6%. But if you increase your density, what happens is this interval disappears. And when you get to the density where we are here, there is no interval. All that means is that the distance between any 2 individuals is going to be short because the density is what it is. What's interesting about the social network is that your 6 handshakes from somebody on the other side of the globe, and it's actually not 6, it's been revised, but it's a very small number of steps. And that is surprising given the large population of the globe and given the very low density, the few numbers of people that you actually know. That's what's interesting. That doesn't exist when you have 67%. In this room, you know, the density of people who know each other here is probably in the region of 60 or 70%. It's not a small world in a significant sense. The small world is to do with sparse graphs, and the cortex is not, the inter-aerial connectivity is not a small graph. It's not a small world. You can come back to the notion of small worlds perhaps in single neurons. Nobody's done the experiment. They could be done with viruses. You could work that kind of thing out, and I think it would be an interesting question. But it's not the same thing. The papers you see with the brain as a small world, and it's built like the World Wide Web. That's how it goes. That's not true. Because what they're talking about, they're not talking about single neurons. They're talking about inter-aerial, inter-regional connections. Now, I said that despite this peculiar density, this high density, there's wiggle rooms for specificity, and it's surprising because when we plot distance with the connectivity, the probability of being connected over distance, it turns out that in the region of a cortical area, well over 90% of the areas are connected, but it's not a flat distribution. As you go further away in millimeters, away from a given cortical area, the probability of it being connected plummets to a very, very small number. So you actually, if you come back to my example, knowing you're connected to some area which is very far distant, actually is a potential source of high selectivity. That's important for the suite, for what follows. One of the findings, and it's a little complicated to explain, but it turns out that if you look at a group of areas like the occipital areas, the temporal areas, the parietal areas, the frontal areas, etc., if you look at each one of these groups and you look at the projections from outside of that group, it turns out that you have a lot of common input. And I'm describing this common input here, where I'm looking at the common input, those areas outside of the occipital lobe which project to every single area in the occipital lobe. That's the common input. When you look at that and you use this newfound versus known connections, it turns out that the newfound projections, because they happen to be long-distance connections, these are the connections which have been missed that I was telling you earlier about, they substantially increase the number of common inputs. For example, here, the frontal eye field. This is the small saccade representation of the frontal eye field, projects to every single visual area in the occipital cortex. V1, V2, V4, the lot. And so that's part of this common input that I'm talking about. So what I want you to look at here, this is the common input, the experimental values. This is the common input that we can see in the prefrontal cortex, the parietal, the frontal, the temporal, and the occipital. The blue bars are what we observe. The common input from the known connections, this is before we did our study, is in ochre color. And the newfound projections are these colors which are in blue. This is the present known and the newfound projections. So that gives us our current status here. And now we're taking all of the connections which are outside of that, and we're making a randomized selection, and we're looking to see how difficult is it to find the particular set of connections which projects to every single one. And it's very difficult to do. What this tells you is that the set of areas is not something by hazard. It's actually a very, very particular set of areas which is providing this common input. That's the specificity of these long-distance connections. It turns out, and we'll come back to this later on, I hope, is that it's often been stated that the prefrontal cortex has a large number of long-distance connections. Well, it does. It's true. We verified it. This belief is actually it's a prediction which comes out of the global workspace hypothesis, is actually confounded, and it turns out, and this will be interesting for the follow-on to this, that the prefrontal cortex also has a very high degree. The number of areas that a prefrontal area connects to is significantly higher, and this will be important for what I have to say later. So now I want to say something about these weight-distance relationships, because I think this is really exciting. One of the bedrock ideas about how the cortex evolves, how the cortex develops, how the cortex adapts, is that you have a necessity to restrict the constraints that the cortex is confronted by. There's a number of constraints. There's the energy constraint. The cortex, the brain, is 2% of the body mass, and it's absorbing 20% of the energy that the body uses. So it's a very, very expensive entity. And minimizing these constraints is recognized to be an important aspect about how the brain evolves and how it adapts. And this weight-distance relationship, I think, is the key to exactly that. So how do we do that? Well, we make these retrograde tracer injections, and we define an index called the FLN. The index, the FLN, is simply the percentage of connections of a particular area with respect to all of the other connections going into a given target area. So I make an injection in area V1. I look at the number of connections in the frontal eye field, 8L, and then I look at all of the connections outside of area 8L, and that gives me a fraction, and that fraction is the coefficient which defines the strength of that connection. And I refer to that as the FLN. So the FLN is an index of the strength of that connection. It turns out, as I said earlier on, that the distribution of weights, if you take an injection in area 10, which is at the frontal pole of the cortex, and you look at the number of areas which project to it, and you rank them by weight, you get this 5 orders of magnitude in the strengths of the connections. And these are the number of areas which project to that. And we've looked at Ken, and I'm not going to come back into that, but it's an important issue in our group, is to look at the statistical reliability of that. And it turns out that we can detect consistently connections across individuals with the weakest connections down at this region here. So if you look at a number of parameters of the way strength and distance interrelate, it turns out that you have a Gaussian distribution of distances. That's exactly what you would expect to have, and you do. And you have a log normal distribution of the weights. And the prediction that comes out of that, the maths that comes behind of that, is that you would predict this decline, this regression. And this regression is looking at how distance and strength of connections. This is looking at the weights of all of the areas at given distances and summing them up and showing that there's this constant weight distribution with this lambda value here. And it's this lambda value here, which I'm going to now show you, is very, very powerful at predicting the geometrical, architectural, and connectivity features of the cortex in general. So I've slightly got a slide problem here. So one way of looking at networks is to look at the motifs. You can take any free areas throughout a particular network and look at different motifs of connection. So in this diagram, you have motifs which go from -2 up to 13. These are all the motifs you can have. You have a motif in which there is no connection, and you have a motif where you have all connections that are possible between these three nodes exist. And you have all these intermediate cases. This way of looking at cortical networks comes out of the laboratory of Allon at the Weizmann Institute, and he argued, and very convincingly so, that the distribution of motifs in any given network gives you an indication of the functionality of that network. And he was able to show that these motif distributions distinguish gene transfer networks, metabolic networks, and neural networks. And his argument is that if we use these motif distribution, we can look at the building blocks of that network. So these lines here are the frequency that we find in our database of these various different motifs. So the most frequent motifs that you can find is the fully connected triangle, and then this motif here, number 12, where you have 2 bidirectional connections and a 1-directional connection here, and then you go down here and you find there's a sort of waxing and waning. Everybody else who's looked at motif distribution in the cortex, and a number of people have, end up with something very, very similar to what we have here. Our data doesn't look different in this respect. What is interesting now is to address the issue. If you want to understand the significance of a particular parameter in the network, what you do is you build a random network and you enforce the particular parameter that you're interested in. So if you take the same number of nodes, the same number of connections, and you make the connections between nodes using a particular rule, you can see if that rule has a significance. So what we do here is we make networks using the EDR rule. That's to say we choose links between areas according to this lambda value that I was mentioning earlier on. And when we do that, the values that we obtain look very similar to the connectivity points, the data points that we have. If we now build random networks, instead of using an exponential distance rule, we use a constant distance rule, so we make random networks. We're not using lambda to build that, then you can see that the values look actually very different. And these differences all add up to being significant. So the EDR rule, the weight-distance relationship observed in the data, generates a motif distribution which is very similar to what we have in that data. This is a complicated slide to go through, but one of the interesting things that we found in the cortical network is there's a huge interest in whether there's a core area or a series of areas in the cortex which function as a hub or a series of hubs. A number of arguments have been made that there's a rich club. The rich club is a series of hubs, so hubs have been defined as high-degree areas, areas which have many, many links. And the observation of Sporns and his colleagues is that if you look at these hubs, you can show that the probability of the hubs being connected is higher than you would predict from the density alone. So hubs showing particular preference to be connected is referred to as a rich man club. Rich man clubs are important in society because you find that people who are on every editorial board, well, they might have a sort of a strong position in the neuroscience field, and they'll be very powerful people. And so this is the rich man club. The rich men are part of every single bank. They're not just all in one bank. They're in all the banks, and the banks are what they are. So we're not looking at rich man clubs because if you look at a rich man club with the density we have, it doesn't make a lot of sense. With these very high density, it doesn't make sense. But what we can do is look at the number of cliques. Now, a clique is a subgraph where every area is interconnected with it. It turns out that we have 17 cliques of size 13, and these are the 17 cliques of size 13. That means all of these areas have, these 17 areas form 13 different cliques, where every single area in that clique is interconnected. That constitutes a core. And this core is shown in a color scheme here, and the areas which are not part of these cliques are in yellow. If you look at random graphs and look for the probability of getting such a high number of cliques, it's 1 in a million. So this is a characteristic feature of the cortical data. It's highly centered in the prefrontal cortex. This fits with what people are finding in the rich men clubs, using collated data, using these low-density graphs, of which I'm so cynical. But basically, they're coming to a very similar sort of finding. Now, I think what's particularly interesting here is that this rich man club is well predicted by the distance rule that I was telling you about. So the black lines are the observed frequency of the high click core in random graphs. The red is what we can predict. And these very high levels here are well connected, are well predicted by the EDR. Without the EDR rule, as I said, you get 1 in a million. It's a very, very, very rare event. I think that result is hugely exciting. It's showing us that a particularly relevant structure, maybe the motives are not quite as relevant as we thought back in 2005, but the core, I think, is going to be very significant, and you can predict it simply by an understanding of these distance-weight relationships, and I think that's uplifting. How am I doing for time? 10 more minutes. Okay, so I'd like to say something about cortical hierarchy, because if you're doing electrophysiology in an area at the moment, you're going to be, I think, probably thinking about the input to that area in terms of whether this is a driving input, whether this is an input which is ascending a cortical hierarchy, or whether it's an input which is descending that cortical hierarchy. This comes back to the David and Van Essen model, which I talked about, 1991, with 2,500 citations. It's probably one of the most represented large-scale models of the brain that we have. So for those of you who are less familiar with it, what it says is you come in at the periphery. This is the retina. This is highly centered on the visual system. You come to the lateral geniculate nucleus, the thalamus. Area V1, you come to V2, and as you march out up towards area 46, areas are staggered on different levels. And this, the composition of this hierarchy has been by making these pairwise comparisons between areas, defining areas as being feedback and feed forward, and making the comparison between them, and then stacking the areas in this logical fashion. And you can see many things in this configuration, which is, of course, very familiar for you, because you have a sort of a temporal Watt pathway, you have a sort of dorsal pathway here, you have the frontal eye field here. And this, I think, has been an extremely important map to the cortex. Now, there are some snags to it, because it turns out Malcolm Young did a search to see if there was a determinate model of the hierarchy coming out of this data. It turns out there isn't. And the reason why is because you have so many of these parallel pathways. So as you're doing pairwise comparisons, whether you put an area above or below becomes indeterminate. So there's 150,000 solutions to the model, the hierarchy, which is the best that we've had up until now. So our best understanding of the cortical hierarchy is indeterminate to a huge extent. Now the data I'm going to be showing you is going to be addressing exactly that. Now, I mentioned that the modern views of how the cortex is functioning goes around two sorts of approaches. The sort of Hubel and Wiesel approach was that information comes into the retina, crawls up to V1 and V2 and higher areas. And in each level, there's some sort of extraction which is going on. And then when you get to the highest level where you've extracted everything and you end up with a kind of a complete process. This is a feed-forward hypothesis, and in some sense, up until the early 90s, it was the best that we had. Because what you could see is that if you isolated or if you knocked out a given level, the subsequent level, the receptive field structure was destroyed. Jean Boulier did an enormous amount of work showing that relationship. So what you could see is that the characteristic response of neurons at different levels was hugely determinant on the connections which were feeding up in this feed-forward direction. So the feed-forward model was a very obvious model to sort of think about. People like Richard Gregory and many others began to question it because they noticed that there was a huge, in psychophysics, there was a huge dependency on what people expected in the environment and what they actually saw in the environment. So when you look at this illusion here, you see something sticking out, and there you see something sticking in, sticking out, sticking in. And if you turn this around, if you put this upside down, you get this. What was out goes in, and what was in goes out. One level of understanding of this, and this goes back to 20 years or so, is that light usually comes from above. And if light is coming from above, and you have this shading, then you interpret that as being convex. And if the light is coming in the opposite direction, in that direction, the shading is in the opposite direction, then you consider that to be concave. These models which come back to this notion rest upon the idea that you have some sort of prediction which is going down the cortical hierarchy, and what's going up the cortical hierarchy is going to be the prediction errors. It's a little counterindicative, and it takes a little bit of getting used to. But it's an idea that comes back from Mumford, and Friston has also been publishing enormously in that. And these ideas have taken gradual hold. So I think cortical hierarchy is important. Now, what is the situation with cortical hierarchy in our data? We can address it, because it turns out that if you make an injection of a retrograde tracer in the middle area, and you look at early feed-forward projections at a long distance away, you'll find that all of the retrograded labeled neurons are in the upper layers. As you come towards, these are successive areas, as you come towards the injection site, the distribution of neurons at hierarchical levels B and C become more evenly distributed. And if you go in the feedback direction, it's exactly the inverse. Long distance feedback uniquely coming from the infragranular layers, adjacent feedback coming from evenly distributed layers. Now, people, when they talk about whole cortical hierarchy, often refer to this sort of connection and this sort of connection. That's what they talk about. And those two connections, I think, are very important, but they're extraordinarily weak. They're very, very weak connections, and I'll argue that they're not driving anything in terms of what we normally understand by the word driving. So using that SLN, I don't know, perhaps it wasn't immediately apparent to you, but as it's a graded value, you can use that to get a measure of hierarchical distance. And when you do that, you can take the Feldman and Van Essen model, which is over here. So this is V1, V2, V3, V3A. This is the temporal pathway, V4, TO, TE, THTF 36. And this is the dorsal pathway, MT, LIP, NST, FST, S7A, and STP. When we do an analysis where we've made multiple injections in these different areas. There's a very, very good correlation between the values that we get for this SLN. I didn't go into the details of it, but believe me. And we get something which looks actually very, very familiar. It looks very, very similar on the two sides. A major difference is the frontal eye field. This is the frontal eye field in Feldman and Van Essen, 8M and 8L. Together that makes up the frontal eye field. If we separate the frontal eye field into the large and small saccade, the large is 8M, the small saccade is 8L. It turns out that 8L is in a feed-forward relationship to V4. It's actually an area, it looks like a cortical area which is very low in that hierarchy, whereas 8M remains at the top of the dorsal pathway. So that's one way of looking at it. This is the only data slide I'm showing. And when we got this about 10 years ago, it kind of blew my mind. I didn't really believe that much in this whole hierarchy business because, quite frankly, it sort of seemed to be kind of overplayed. What this injection is an injection in area V4, and an injection in area V1, and then looking at area V2. So you remember V1 is a lower area, V4 is a higher area. We're simultaneously looking at the two sets of neurons which go to the higher and go to the lower area. And when you do that, it turns out that they're highly segregated. And what I expected is for everything to be mushy and uninteresting and boring and get actually neurons in area V2 which project both to V4 and to V1, and you don't. And you don't. And I found that hugely, I mean, that sort of made me very, very interested in hierarchy because that's unusual. If you do any other injection, you get two populations that are going to the same place, they show bifurcation at rates of about 30 or 40%. There's a huge amount of bifurcation in the cortex. So this is very, very, very unlikely. Now, if you look at this, there's a pattern here. There's a pattern. There's a very definite pattern. This is looking at V2. So the red dots are going up to V4, and the black dots are going to V1. And you can see that there's not very many double-labeled neurons. They're indicated by stars. I think there's 2 here. They're usually actually in the infragranular layers, which I think is very interesting, but we won't go into that. But there's a vague segregation here. It's not actually vague, it's very clear. It turns out that the feedback connections in the upper layers come out of the top of layer 3, and the feed forward in the upper layers come out of the bottom of layer 3. And yes, the neurons in the bottom of layer 3 target layer 4, which is what they're meant to do, and the ones from the top of layer 3 don't target layer 4. They project to the top of layer 3. And that, I think, is very interesting. This is not layer 2. Layer 2 is a layer which is just underneath this V1, and it was referred to by anatomists at the beginning of the last century as the outer granular layer, which I think is also intriguing. So when we put this together, what we get is you have a counterstream organization. You have a counterstream organization where, yes, you have a long-distance feedback coming out of the infragranular layers, and you have short-distance feed forward in layer 5. That's sort of not a very, very pronounced feature. But what is this pronounced feature is this long-distance feed forward. This is the classical feed forward from the bottom of layer 3, and then a short-distance feedback from the top of layer 3. This counterstream organization, you find it throughout the whole of the visual system. And again, if you go back to Tiggers and Tiggers in the early 70s, you can see it. They sort of cite, you mention it, it's in HRP, and it's not really pointed out. And this notion of a counterstream, I think, is very interesting, and it was an idea that Simon Ullmann was pushing a lot. He was pushing this idea a lot in the 90s, and I think it's highly significant. So I'm gonna wrap up, and I'll make my conclusions. Now, this is looking at V1, V2, V4 projections. When you look at the distribution of strengths, this is the strongest connection that's color-coded. So V1 is green, and you go from the projection from V2 to V4, and these are all the connections that you get coming into area V1. And then V2 is like this. So this is this log-normal distribution that I was telling you about. This here, this star here, is the, this is color-coded. You have You see, you have this little arrow, right? So that's the lateral geniculate. That's the projection of the lateral geniculate nucleus into area V1. Now, it's been hugely studied because that projection actually provides something like 5% of the synapses in layer 4. And it's driving, right? I mean, if something is driving in the brain, it's the lateral geniculate input to layer 4. But it only just does it. I think that we can make a good argument that everything below that line is not driving. I think that argument is pretty easy to make. If you look at the coupling, the physiological coupling between lateral geniculate axons and layer 4 neurons, it's very, very particular. So that and you look at the morphology of the synapses, the size of the synapses, the synaptic thickening, there's a lot of features that says these are powerful inputs, but the strength of it is actually very small. And I'll give you that the connections which are below that line are not driving. And most of those connections are actually, a lot of them are connections that you're very familiar to if you were sort of studying it. So if I was to come back and say, put all this together and look at the David Van Essen, Feldman, Van Essen hierarchy, what does it say? Well, I haven't done this, but if I was to draw in the driving connections we're finding, they're to mainly adjacent levels. At most to adjacent levels. There's actually one exception with V1. That seems to sort of drive up to a bit further. But mostly between adjacent and lateral levels. So this notion of driving and modulating, which I really don't think is interesting at all, but if you really want to hang on to it, I don't think it distinguishes feedback from feed-forward. It distinguishes these broadband connections, which are actually really very, very, very local. So I think we have a mystery about what these long-distance connections are actually doing. They're highly specific. They're very, very prevalent. There's a lot of them. They constitute a dense network. And I think what they're doing is they're coordinating activity across the cortex. So this is a summary. This is what I've tried to tell you. Interareal connectivity is highly asymmetrical. Don't let anybody tell you that it's bidirectional. It's not. That's because they're working with DTI and they can't say anything else. The graph, the cortical graph is very dense. 30% of the connections in that graph have not previously been recorded. There is not a small world in the interareal cortical network. There's a huge importance of weak links, and I think we need to go back and look at this Granvetter. He was about the same time as Milgram. We were talking about Milgram the other day. He's in that sort of class of impact in social networks. Weight-distant relationships are hugely important. They allow a prediction, and that prediction is important because it tells you that this minimization of wire, which has been the major adaptive feature which people have been looking at, is working through this exponential distance rule. The cortical hierarchy definitely exists. It's a powerful idea, and I think the counter streams is going to say a lot about it. And modulating versus driving, I don't think is going to be a useful distinction on large-scale networks in the future. Thank you very much for your attention. Do you have time for a question, or is that it? Yeah, I think that's a very interesting question. The dogma is that inhibitory cells are local, that they don't project over large distance. Of course, that is a dogma, and there are inhibitory cells which are projecting over some distances, and I think it would be very interesting to make that distinction. We're not doing that, but it's certainly something that one would like to do, and I think the way you would do it is you would come back over our sort of data, or that kind of retrograde data, and just do stains and see how many smooth cells, what is the proportion of smooth cells. It will be very, very small. No, because the bulk of the inhibitory cells are going to be much shorter distances than what we're having. Yeah, no, it won't have a big effect on that local versus global connectivity. Well, so the clicks are very centered in the prefrontal cortex. They're not uniquely in the prefrontal cortex. The areas we've defined are only, if we increase the number of injections, we can find more clicks, we won't find less. So what we have there is correct. And they're being centered in the prefrontal cortex makes sense with the fact that the prefrontal cortex does have an excess of long-distance connections, and areas in the prefrontal cortex connect to many, many more areas than, say, in the parietal or occipital cortex. So I think that the fact that you have long-distance connections and the fact that these clicks, so they're going to make destruction of the click will have a big effect on the communication across the cortical network. So it shows you that the prefrontal areas are going to be playing a key role in activating large regions of the cortex. All those peripheral areas are feeding through the clicks to get to other areas. So the function of the rich club or the function of the clicks is going to be hugely on promoting the communication through far distance points. So they could be, you could think of them playing a role in global workspace kind of activations of large spaces of cortex.
I have my usual question about possibility of cortical talent in key that is shorter for your graph.
Right. Well, that's also an important issue, and we need to know we need to know something about what are the numbers of neurons involved. Some people, like Murray Sherman and Ray Guillory, feel that actually cortical-cortical connections are really of no importance, that we can forget about them and go home and get on with other things, or rather forget about them and go home and think about the thalamocortical loops. I think they are very important, obviously, but I don't think they're going to be hugely involved in long-distance communication in the way that some people would hope. So they will be affecting long-distance communication, but they're not going to be broadband in the sense that these short-distance connections which I was talking about earlier on, the strong connections which exist in the cortex are not going to be extended. You're not going to have communication between area V2 and area 46 via some thalamocortical loop. You're not going to have anything better than what you get, I think, than going through the cortical-cortical loop, going through the cortical-cortical pathway.